Now moving on to the next question, that is question number 7. How did Hershey and Chase differentiate between DNA and protein in their experiment while proving that DNA is the genetic material? Alright, these were the two scientists, Hershey and Chase were the two scientists who cultured bacteriophages in two different medias. These both scientists cultured bacteriophages in two different medias. One media is containing radioactive phosphorus and the another one media of bacteriophages containing radioactive sulfur. Now, these scientists allowed these bacteriophages to infect their host cells, that is Escherichia coli. Now we know that phosphorus is the content of nucleic acid, is the content of DNA which forms the nucleic acid, and the sulfur is the content of amino acids such as methionine and cysteine, which are the amino acids among 20 amino acids which forms protein. Now after infection, they take the samples for analyzation and they separated these bacteria and viruses with the help of centrifugation. Now we know that bacteria are larger in size, so they formed a solid pallet at the bottom after centrifugation, and phosphorus or the viruses remained in the supernatant of the centrifugation. Now after analyzation, the host cells or bacteria are found to be radioactive when they infected with radioactive phosphorus, whereas no radioactivity was found in the host cells or Escherichia coli or bacteria which was infected with the radioactive sulfur. This proves that the DNA is actually the genetic material. Now let's write the answer of this question number 7, and it will start like this, that Hershey and Chase, these were the two scientists who cultured the bacteriophages in two different medias, One is containing radioactive phosphorus, and the other one is containing, the other media is containing the radioactive sulfur, Now we know that phosphorus is the content of DNA, and sulfur is the content or constituent of amino acids such as methionine and cysteine, which are the amino acids among 20 amino acids which forms protein. Now the scientists such as Hershey and Chase allowed these samples or we should not write samples. They allowed these bacteriophages, which are viruses, bacteriophages, to infect the host cell, that is E. coli. That is E. coli, which is a bacteria. So they allowed these bacteriophages, which are the virus infecting bacteria, to infect the bacteria such as E. coli or Escherichia coli. After infection, for analyzation, they separated the bacteriophage or viruses, bacteriophage and E. coli, in which the E. coli forms a solid pellet at the bottom, and viruses, because they are smaller in size, viruses remained in the supernatant, right? Now, after observation, what they found, that the bacteria or the host cell, bacteria or host cells are radioactive when infected with the radioactive phosphorus, and no radioactivity was there in the bacteria or host cells when infected with the radioactive sulfur, which proves that DNA is the genetic material. And on the screen we have a written solution for this question. Who find difficulties in writing the answer, long answers in English? This written solution is for those students. In this solution also, the same thing is written, that Alfred Hershey and Martha Chase, which is the complete name for these two scientists, they cultured bacteriophages in two different medias. One is containing radioactive phosphorus and another media is containing radioactive sulfur. Now, we know that phosphorus is the component or content of nucleotides, which are DNA, which forms a nucleic acid such as DNA, while the sulfur is the component or content of amino acids such as methionine and cysteine, which are the amino acids among 20 amino acids which helps in the formation of protein. Now, these two samples of bacteriophages were allowed to infect their host cells such as Escherichia coli or E. coli, which is a bacteria. Now, after infection, the samples were analyzed for the radioactivity in the host cells, of course. For analyzation, what they did, they separated the bacteria and viruses via centrifugation. Since the bacteria are large in size, they form solid pellet at the bottom of after centrifugation, and smaller viruses remained in the supernatant after configuration. The bacterial pellet was found to be radioactive when infected with the radioactive phosphorus or the bacteriophages with radioactive phosphorus, and there was no radioactivity found in the bacteria or Escherichia coli which were infected with the bacteriophages with radioactive sulfur. Now this things proves that the DNA is actually genetic material. DNA is the genetic material, not proteins are the genetic material. And on the screen you can see a diagram. This is the experiment 1, this is experiment 2. In the experiment 1, we are testing the proteins. We are testing proteins with the help of radioactive sulfur. Now see, this is a bacteriophage, a virus which is able to infect any bacteria. So this is a bacteriophage. The scientists radio-abled the protein codes of this virus and allowed this bacteriophage to infect the bacteria such as E. coli or Escherichia coli. Then they analyzed it and no radioactivity was found in the Escherichia coli when it was infected with the bacteriophage which was labeled with radioactive sulfur. But in the experiment 2, when these scientists radio-abled the DNA of this virus or bacteriophage and allowed this bacteriophage to infect the Escherichia coli or bacteria, after analyzation they found this Escherichia coli or E. coli was radioactive. Alright, so from this experiment they proved that genetic material is DNA but not proteins. This is a complete solution of question number 7.